CEGS is interested in saying, if we have a specific piece of DNA, what proteins are bound there? And can we identify all of the proteins on a specific piece of DNA?
So in order to make this complex technology work, we basically need technology developments and improvements in 4 separate areas. The first part is we need to come up with efficient ways of cross-linking DNA binding proteins to the DNA so that they stick on there. Then we need to be able to break up the DNA into smaller pieces that we can actually handle. So that's the first part, the cross-linking and the fragmentation and getting these pieces out of a cell.
We are trying to develop technology and actually have been very successful at this point in developing technology that we can scale down to the level of single genes and cells to be able to interrogate protein DNA binding at gene regulatory regions at the genome level.
To really work on developing new methodologies to process these complex yeast cell lysates, because yeast is our model system, and figure out how, basically where the DNA protein complexes that are cross-linked to each other are going.
The second step then is, how do you capture now one particular DNA sequence on a surface or on a bead in a sequence-specific manner so that you know which piece you've pulled out?
Our goal is to be able to do sequence-specific capture, in other words, based on a particular gene sequence that we know, because we know the human genome sequence. We want to be able to go in and fish out that segment of the genome and also pull out with it the proteins that are interacting with that segment of the genome.
We've been interacting a lot with Lisa Cirillo's group in Milwaukee, and she's produced a number of protein DNA complexes for us to take back here and to do analysis on in efforts to achieve the milestones that we've set for ourselves.
Technology development on. The next part then is, once you have captured your piece of DNA, you want to identify all the proteins that are stuck to this piece. And that the technology for this is mass spectrometry, which has been used extensively in proteomics, but not necessarily in the approach that we are trying to use it with, so that you try to identify proteins off a surface.
We isolate proteins from particular regions on the DNA, and then we analyze them via mass spectrometry.
We also then need a very powerful analytical tool called a mass spectrometer, which will allow us to identify and eventually quantify what proteins are present on those minute amounts of material.
And then the last part, once you have the mass spec data, then you need to develop the appropriate analytical tools that allow you to look at this, help you identify protein complexes or how they're modified. And while some of these tools are available, they have never been used in an efficient manner in this context of DNA binding proteins, so there's likely to be additional tools that we will need to develop to make that work.